Have you ever wondered if two male mammals could have a biological child together? Well, in 2025, a team of Chinese scientists made history by doing just that. They raised a mouse, born from two male parents, all the way to adulthood. And the secret? A radical rewrite of the genetic imprinting that usually blocks such a possibility. Let's explore how they pulled off this feat, why it matters, and what it could mean for the future of reproductive science. In mammals, both a maternal and a paternal genome are needed to create life. That's because certain genes are imprinted, meaning they are turned on or off depending on whether they come from mom or dad. When both genomes come from the same sex, things fall apart. Embryos fail to develop. For bipaternal embryos from two sperm, the failure happens even earlier than with two maternal genomes. Li and his team zeroed in on this barrier. In previous efforts, correcting 7 key imprinted regions wasn't enough. The embryos looked abnormal, swollen bodies, protruding tongues, hernias, and they quickly died. So the team asked, what if we modified not just 7, but 20 imprinting sites? They first created 18 KO mice with 18 deleted or modified imprinted loci. These mice showed dramatic improvements. The organs looked normal, and the newborns could breathe on their own. Yet almost all of them still died, not from organ failure, but from something as basic as an inability to suckle. The researchers noticed that a specific imprinting region, the Nat-Blay-Cap locus, was still dysregulated. This region affects craniofacial development, which in turn affects suckling. They deleted it, creating what they called the 19 KO mice. The result? Mice that could suckle, grow, and survive. With 36.7% of these mice surviving naturally, the results were stunning. Even with 19 imprinting corrections, natural pregnancies with bipaternal embryos still failed early. Why? The answer lay in the placenta. One imprinted gene cluster, CEFMT2, wasn't corrected in 19 KO mice. It includes 72 microRNAs critical for placental development. So they created a 20 KO line, deleting this entire region. The result? Fully developed mice with functional placentas, the missing piece to natural development. But could these mice reproduce? Not naturally. Female bipaternal mice couldn't conceive, likely due to the very genetic modifications that saved them. However, using fibroblasts from these 20 KO mice, the team successfully cloned new offspring. These mice weren't just healthy, they behaved differently. 19 KO mice were less anxious, grew faster, and had reduced hippocampal volume. This work doesn't just open doors for same-sex reproduction. It rewrites how we think about developmental biology, genetic imprinting, and stem cell potential. But it also raises profound ethical questions. If we can build a mammal from two males, what else might we try? And should we? The researchers suggest that instead of permanent deletions, future work may use epigenetic editing, turning off genes without removing them, to avoid the risks seen in these mice. The birth of a mouse from two dads is not just a scientific milestone. It's a leap across a fundamental barrier of nature, made possible by manipulating the most mysterious elements of genetics, imprinted genes. We now stand at the edge of what's possible, where genetics meets engineering, and where even nature's oldest rules can be rewritten. Like, subscribe and tell us, should science keep pushing these boundaries? Or are there lines we shouldn't cross? Let us know in the comments.